The liver is the largest gland in the body, weighing about 1.4 kilograms in an adult. It is situated under the diaphragm in the upper abdominal cavity and is held in place by several ligaments. It is a reddish-brown colour and comprises of 4 anatomical lobes. When viewed from the front, the dominant left and right lobes can be seen, which are separated by the falciform ligament. Situated in a depression on the posterior surface of the liver is the gallbladder, a pear-shaped sac which stores bile synthesized by the liver. The liver performs many vital metabolic functions. It has the ability to store and metabolize useful substances such as nutrients, but it breaks down or detoxifies harmful substances to render them inert and less harmful. The liver receives a blood supply from 2 sources. The first is the hepatic artery, which delivers oxygenated blood from the general circulation. The second is the hepatic portal vein, delivering deoxygenated blood from the small intestine containing nutrients. The blood flows through the liver tissue to the hepatic cells where many metabolic functions take place. The blood drains out of the liver via the hepatic vein. The liver tissue is not vascularized with the capillary network as with most other organs, but consists of blood-filled sinusoids surrounding the hepatic cells. The liver lobes are made up of microscopic units called lobules, which are roughly hexagonal in shape. These lobules comprise of rows of liver cells or hepatocytes which radiate out from a central point. The hepatic cells are in close contact with blood-filled sinusoids and also lie adjacent to canaliculi into which bile is secreted. Situated around the perimeter of the lobule are branches of the hepatic artery, the large hepatic portal vein and the bile duct. These cluster together at the corners of the lobule, forming what is called the portal triad. At the midpoint of the lobule is the central vein. Blood flows out of the sinusoids into the central vein and is transported out of the liver. The hepatic portal vein and hepatic artery deliver oxygen and nutrients into the blood sinusoids. This close relationship between the hepatocytes and surrounding blood enables many metabolic processes to take place. Blood flows out of the sinusoids into the central vein, removing detoxified substances and metabolic end products. The central vein ultimately reunites with the hepatic vein, transporting these substances out of the liver. Bile that is produced by the hepatocytes drains into tiny canals called bile canaliculi. These drain into bile ducts located around the lobule perimeter. Hepatocytes are the predominant cell type in the liver. An estimated 80% of the liver mass is made of these cells. The hepatocytes are round in shape, containing a nucleus and an abundance of cellular organelles associated with metabolic and secretory functions. Organelles include endoplasmic reticulum, both smooth and rough, and Golgi apparatus for secretory functions. There are also high numbers of mitochondria to provide energy to support the many metabolic functions of the liver. Some of the hepatocytes lie adjacent to endothelial cells which form the walls of the sinusoids. These 2 cell types are separated by a small space called the space of Dis. Attached to the right lobe of the liver by connective tissue is the gallbladder. Shaped like a pear, it is 7 to 10 centimeters long. Bile produced by the hepatic cells drains out of the lobules via the bile ducts. These unite to form the left and right hepatic ducts, which join the cystic duct, transporting bile to and from the gallbladder. The gallbladder concentrates and then stores the bile until it is required to assist in the digestion of fats. When needed for digestion, Bile travels through the cystic duct into the common bile duct. This unites with the pancreatic duct to empty bile into the duodenum. Bile flow is under the control of the hormone cholecystokinin and enters the duodenum through the sphincter of Oddi.